Good afternoon, everyone. I've often been accused by my colleagues of my dispassionate communication or about the pipeline, dispassionate but objective. So I'll try to amp up the passion, but I will be objective. What I'm going to talk to you over the next half hour or so is I'll walk you through each of the programs. I'll talk a bit about the indication. I'll spend a little bit of time talking about the insight and then how we validated the insight. And then I'll spend the majority of time for each program talking about the trials with really an emphasis on what are the expectations, what does good look like. So with that, let's get into the pipeline. So this is the pipeline. This is a robust pipeline. As you can see, we have 4 ongoing phase 2 studies. We have a phase 2 study that's starting in Q4 of this year for our C. diff program. And you heard a little bit about the INDs. The RBM39 program Laura mentioned earlier. We expect to submit that IND in Q3 and initiate the study in Q4. The Epsilon program we plan to submit early in Q1 of 2025. So what you heard about this morning was how the company is rapidly translating insight into clinical programs. And this is the evidence of that here. This is a monumental amount of work. We expect that this pipeline will increase. But what it seeks to achieve over the next 18 months are 7 readouts of both phase 2 and phase 1 data. So with that, let's get into the first program. So this is our REC-994 for the treatment of symptomatic cerebral cavernous malformation. So this is probably the largest disease that no one has heard about. It's certainly a disease that I was unaware of before I came to Recursion. So believe it or not, there are over 1 million patients that suffer from this disease. About 1/3 of them are symptomatic. Symptoms can range from mild headaches to severe hemorrhagic stroke, right? So there are no approved therapies, and there's been only one completed phase 2 trial run by a relatively small investigator-initiated group. So most patients are either observed, their symptoms are managed through over-the-counter meds, or they're offered surgery, in some cases radiation therapy. But that's really dependent upon a lot of things, right? How bad are the symptoms? Where are the lesions? If these are deep lesions, it's going to be really, really hard to go after. So the unmet need is significant. At the heart of the pathology is altered endothelium that leads to vascular malformations. Our therapy targets the endothelium. So a question that I've heard, and Michael has heard, and others have heard, is if this is such a problem and there are so many patients, why aren't more companies going after this disease? Well, look, I can't speak for other companies, but I can kind of speak to from a biologist perspective, and I sort of pretend to be a biologist at some time in my life. You know, the biology here is very complicated. You have multiple proteins interacting, converging on multiple pathways. So the target is unclear, right? These are the types of biology and indications that lends itself to the Recursion OS, right? So we can go after, in an agnostic approach, go after looking at many different molecules and try to come up with the one that fits best. So this is a program that has sort of founded in the roots of the company, and we're very excited about the readout coming in Q3 of this year. So I've talked a little bit about the insight, right, where this came out of. This is some preclinical data that's been around for a while. This came out of our founder's PhD dissertation. And it covers a couple of panels. The top panel looks at 2 CCM knockout models. And what you can see as compared to control, we're able to reduce the lesion burden. So some of these patients have multiple lesions, right? 5, 6, 7, and others have few. But in both cases, both the size and the lesion burden were reduced. We think perhaps what's more important and might be clinically relevant is shown on panels 2 and 3. We think inherently this is a disease of the vascular permeability. And we think that our therapy, at least in these models, showed that we're able to impact the permeability. So this is something that we can monitor, measure in the clinic. So this is the study design. So we've enrolled, and this study happened to enroll relatively fast. I think we were able to enroll 60 patients in somewhere around 9 months or so. So really speaks to sort of the unmet need and the desire for patients to get treatment. So all patients will have MRI-confirmed lesions. There is a familial component of this disease, which accounts for around 20 to 30% of the patients, and then there is a sporadic form. We're enrolling both patients. All patients need to have symptoms that the clinician felt were directly related to CCM. So these are not patients that are walking around that are asymptomatic. I'll talk a little bit about the primary and secondary endpoints in a minute. You can see the schema. It's a randomized, blinded study. Patients are treated for a year, and then they're given the opportunity to go on the extension study. Most of the patients, I think to date, there may be only 3 patients that have not moved on to the extension study. So by and large, patients are continuing on study, and we will follow those patients as they progress in their therapy. So why a year, right? Why not 6 months? Why not 3 months? Well, this is a slow-growing disease. Patients often live with this disease their entire life. So input from KOLs, it was felt that 12 months is sort of the minimum amount of time that you would want to treat and treat these patients. So obviously, we're going to be treating these patients. We will continue to treat them in the extension phase, and we will be monitoring their therapy. What are we looking for? This is the question that we get asked again and again. So this is not a response rate study. This is not a PFS study, right? The primary objective of this study is safety and tolerability. And when I've talked about the primary being safety, I get a lot of eyes rolling. Yeah, who cares? It's important, right? It's important to not only have a therapy that's effective, but it's important to have a therapy that patients can take for a long time. An effective therapy that patients can only handle for 6 months is not going to be one that's used, right? So we want to be able to evaluate the safety of these patients for a long period of time. Now let's talk about the secondary and exploratory endpoints. So why wouldn't you elevate efficacy to a primary endpoint? The answer is we simply don't know enough about the disease. We don't know enough about it. KOLs don't know enough about it. The FDA doesn't know about it. There's been no real data to say what efficacy endpoint is important. So we're not going to sort of fictitiously power a study around an endpoint that we have no data. So this, in essence, is an exploratory study. But what are we looking for, right? What does good look like? Well, it's shown here, really. Our analysis is going to be looking for multiple trends across multiple endpoints. I've talked about permeability, looking at hemosiderin deposition. I've talked about changes in lesion burden. Subgroup analysis is going to be really, really important because this is an inherently heterogeneous disease. You have some patients that have come on study that have had hemorrhage. You had other patients that have come on study that have big lesions, others small lesions, others with headache. So I think it would not be prudent for us to just sort of go after an efficacy endpoint. But what we're trying to find are trends that are meaningful for us, trends that are meaningful for KOLs, and most important, for the patients. Our hope is that if we see these compelling trends, we're going to be able to make a case with the FDA on what does your next study look like, and whether that be a 2-3 or right into a 2-3, right into a 3. So I think we're very excited. The team has done a lot of work. This has been a well-run study. Investigators and patients have been very passionate. And I just want to acknowledge Connie Lee, who leads the patient advocacy group for CCM. Any questions about CCM? Ask her. She knows more about it than me. So our next program is our 2282. This is our HDAC inhibitor for the treatment of progressive NF2 mutated meningiomas. So a little bit about the disease. So NF2 meningiomas sit within a constellation or a syndrome known as NF2-related schwannomatosis, right? So within that disease, you have meningiomas, schwannomas, ependymomas, meningiomas. It's a complicated disease, and not all the tumors are alike. You can just imagine what this disease is. Patients suffer from multiple tumors that are benign. The standard of care for the treatment of these patients is surgery or radiation, but this is a relentless disease. You cut out the tumor, it comes back. You radiate the tumor, it comes back. You shrink the tumor, it comes back. So clearly, a therapeutic option for patients who don't have to go through 7 surgeries like this patient has is critically important. I'll talk a little bit about sort of the endpoints. What the FDA has communicated and what the field has tried to show was that you really want to delay progression. So we'll talk about sort of the endpoints that we'll be evaluating in our study. So you've seen the panel on the right. I'll just briefly frame it up as insight on the right or on the left, validation on the right. So we were not looking to build an NF2 program. This is insight, novel insight that came out of our platform agnostically. And you can see the validation in what is considered a clinically relevant FAP model. Rather striking, prevents growth, and also what's probably most important is the regrowth, right? Again, these tumors come back, so simply just decreasing their size is only half of the equation. You not only have to manage the tumor, but you have to prevent regrowth. So, this is fairly compelling data, along with all the other work that we've done. We've translated this into our study. So there have been a lot of molecules that have been tested in NF2 disease, right? I think one of the challenges that these clinical trials have faced is the heterogeneity of the disease. So this is a disease that waxes and wanes. Patients have progression, and then they become indolent. Patients have progression, and then they become indolent. And you're also mixing different types of tumors together in a trial. So what we've tried to do in this study is control for that, right? So we're only evaluating meningiomas. Obviously, if we see a signal, it'll give us reason to believe that we might be able to go after other tumors. And the other thing that I think is probably more important is we've tried to control for progression. What you don't want to do is enroll patients that have relatively indolent disease, and then it's difficult to interpret their progression-free survival. Is it just because the patient simply doesn't have aggressive disease. So what we tried to do, and the advice that we've got from our KOLs, is try to enroll patients that have more aggressive disease. So these patients will have had to progress over the last 2 years, so we can kind of understand their slope of progression prior to coming on study. And obviously, what we're trying to do is decrease that slope of progression. So these are one of the endpoints that we'll be looking at. We'll also be looking at, you can see a futility analysis of PFS6. We've enrolled patients that have sporadic disease and familial disease. We'll be evaluating them together as well as in separate cohorts. So essentially, we've tried to control for things that other trials have not controlled for. You can see that they're dosing across 2 different dose levels. We'll get to this PFS analysis. We expect to have top-line data by the end of this year. If the data is encouraging, then the FDA said, come back and we'll talk about the next part of the study, which would likely be a controlled study that is randomized. So stay tuned for this data. Our next program is our MEK inhibitor. So this MEK inhibitor is being evaluated in 2 studies. One is an FAP study, which I'll talk about here. Another is our study in Axon 1 APC, which Najat mentioned. So I hate to sort of characterize diseases as bad or worse, or this is a tough disease. Most of the patients that are diagnosed with this disease are diagnosed at a relatively early age. Often in their teens, it's an autosomal dominant. This is characterized by, if you can imagine, hundreds upon hundreds of polyps in the colon, right? Just imagine that. Patients are typically diagnosed in their late teens, early 20s. The treatment of choice is colectomy. So imagine having that as a 20-year-old, right? The risk in patients that are not resected, the risk of cancer is 100%, right? So even though you remove the colon, you still have polyps throughout the small intestine. These patients need to be monitored throughout life. And they're often managed through surgery because there are no approved therapies. So it's unfortunate, but over time, you're having more surgeries and patients are becoming more restricted. There's more morbidity. It's a horrible, horrible disease. So really what we're trying to do in this study is take some of the insights that came out of our OS, similar theme, and validate them. So this is another clinically relevant model, and I'm just showing you one slide. It's late in the day. I don't want to bore you with 1,000 slides, but this is fairly impressive data. In comparison to controls and other drugs that have shown, celecoxib has shown a decrease in polyps, we have a nice dose-dependent impact on both polyp count and high-grade dysplasia. So again, compelling data that we simply can't ignore. The study is as follows. Because this molecule has not been evaluated outside of cancer patients, we felt that it was particularly important to take a conservative approach fairly early on in the study. We really don't understand what the removal of a colon might have on the PK. So what we're doing here is a short dose escalation study. And once we have the recommended phase 2 dose, we'll expand it into a dose expansion. So what does good look like here? I think for us, what good looks like is a reduction in polyp burden. Can we shrink the adenomas? Can we overall reduce the number of adenomas? We think this is an important signal-seeking study. If we can show activity, then I think there's reason to believe that we could move this forward. But I think this is a This is a sort of a succinct way to design a study and really understand whether or not if you have a drug that you can take further. So this study has been initiated. We are enrolling patients. We expect to have preliminary data by the first half of 2025. Our next program is with the treatment of solid tumors with AXIN1 or APC mutations. So if you look on the right, people have known about AXIN1 APC for years. This is not anything new. AXIN1 APC sit in a cluster of proteins that regulate the Wnt pathway. And people have known that there have been mutations within these genes for years as well. But it was always felt to be an undruggable target. Until you can really drug the target, you really don't know the clinical relevance of AXIN1 APC mutation. So right now, a patient comes in, they're screened with NGS, they have an AXIN1 APC mutations. There's nothing that the physician can do about. They'll go on standard of care. But we think our insight here is really novel, and there have been MEK inhibitors around for a long time. People know a lot about them, but this is really, unique insight that came out of Recursion. So here you see sort of the phenotypic screen, and what you found is that this particular MEK inhibitor, and it's this particular MEK inhibitor, not a class effect amongst all MEK inhibitors, was really phenotypically opposite to AXIN1 and APC mutation. So, okay, interesting, right? What do we do with that, right? Well, let's try to validate it in some models. And here you see on the right, we've looked at the relationship of targeting AXIN1 and APC mutations versus wild type. And we see a statistically significant improvement in both tumor growth inhibition as well as progression-free survival. So again, another insight that we think that we know is unique to Recursion. The trial is a fairly standard Simon two-stage design. We're enrolling patients with AXIN1 APC. We'll do a quick safety run-in at 12 mgs, and we're looking for response rates. If we see one or more out of the first 10, we will continue to enroll. Najat alluded to some of the approaches that we've taken to enroll patients. I think we have a very aggressive strategy, and this study is enrolling briskly. So this study is ongoing. We expect to have the phase 2 readout in the first half of 2025. So you start to get a theme, right? Lots of readouts. We've talked about it a lot, but this is a pipeline that is producing, hopefully, meaningful and positive clinical data. Our next program is with our toxin inhibitor, C. diff. So I don't need to explain to anyone in the room that C. diff is a problem. It's a problem here. It's a problem worldwide, right? Patients die from C. diff, right? This is not something where you just simply throw antibiotics at it and it goes away. And in fact, probably about 30% of the patients, 20 to 30% of the patients recur. And not just recur once, right? Recur twice, 3, 4, 5 times, right? So optimal treatment to C. diff should really factor on 2 approaches. One is to kill the bug with antibiotics, and that is fairly effective. The other is using an antitoxin, right? Because it's the toxins that produce the pathology, it's the toxins that produce the symptoms, and it's the toxins that predispose patients for further recurrence. This is our first NCE that we've taken to the clinic. RBM39 hopefully will be our second. Epsilon will be our third. Starting to see a theme, right? So the study, so this is some preclinical data. Again, insight on the left. But I think this data is fairly compelling on the right. So this is a well-known C. diff hamster model. Hamsters are infected with C. diff. They're treated with, in this case, 4 options: vehicle control, vancomycin, or in this study, our antitoxin versus the only other antitoxin that's approved, bezilotoximide, which is an IV infusion. So as you would expect, vanco does really well, surprise, surprise. But I think what everyone can see is a rather striking and significantly difference in probability of survival between our molecule and other antitoxins. So this is a program that is gathering a lot of interest among the community. Patients that have at high risk or have recurrent disease are often given antibiotics, and they're offered either a fecal transplant, which is not often readily available, or bezilotoximab. So we think there is an opportunity to impact here. The trial is straightforward, right? So we've taken patients that have high risk of recurrence. There's a fairly standard algorithm to identify those patients. They'll be treated with standard of care vanco. My sense is that most, if not all, of the patients will have symptom resolution. Then they'll be randomized to high, mid-dose, or observation. We expect that patients in the observation arm, based on real-world data, should start to recur within 4 to 6 weeks. So what we're trying to do is delay and improve on the rate of recurrence. So this study is expected to start in Q4 of this year, and we're very excited and hopefully have preliminary readout by the end of 2025. I'll quickly just walk you through, you've heard a bit about our RBM39, another NCE. This is the study design that's hopefully starting up by the end of the year. Fairly straightforward, phase 1, and then we'll get into dose confirmation. So this will be a study in biomarker-selected, relapsed, refractory, HR proficient solid tumors. We have not given any guidance on what the biomarker selection criteria is. As we get closer to the study, we will be providing that information. But again, we'll be using a lot of the same tactics to enroll patients, and we expect that there'll be a lot of enthusiasm. Hopefully, this study will enroll quickly. So let me switch gears a little bit. I'm not going to walk you through this slide. You guys have seen it. It's even too complicated for me. But look at it at its heart, right? The essence of what this is all about is moving really rapidly from insight to IND. So when I interviewed here, Chris Gibson sort of challenged me. He says, well, what are you going to do to be innovative? We're innovative. Look at all this great stuff we're doing. Clinical development, it's a wasteland. There is no innovation, right? It's where efficiency goes to die. That's clinical development. So I said, Chris, stop bothering me. But more importantly, clinical development is less about innovation, in my opinion, and more about efficiency, right? There are a lot of tools out there, right? We cannot take 6 months to go from IND to first patient, first visit, right? And it's less about speed, right? We are regulated to go at a certain speed, but it is about efficiency, right? So what are we doing, right? So Chris is often challenging me, what are we doing to be more efficient? So, I mean, here you see just a few of the steps, right? So, look, I have to be efficient, because as the recursion engine starts to pump out molecules, if I haven't taken a more innovative, efficient approach, there's going to be a backlog. And the clinical development program will sort of die in a quagmire, right? So some of the things that we're trying to do to make things more efficient, right? Look, One of the big problems that we have in the IND process is a very long GLP tox. It takes a month to run this study. It takes 4 months to get the data, right? So that's 3 months where we're sitting around not doing anything. So Recursion is constantly trying to think about ways that we can, I don't want to say get around it, but use our models, use our in silico testing that maybe start to move away from traditional GLP tox studies. Obviously, this is a conversation that we need to have with the FDA, but it's an important conversation. It's not one, we can't do standard practice. It's not going to work here, right? Manufacturing is rarely talked about, but it's one of the biggest black holes of money and time, right? The traditional approach is really built around making very large-scale amounts of drug, holding it, right? That takes a lot of time and effort. What we're thinking about and what we're doing is making quicker, smaller batch that can meet the needs of a trial within months of when we start the trial. We don't have to sit around. I've been in many trials in my life where I've sat around for 6 months waiting for drug supply. That's not going to happen here at Recursion. Biomarker patient selection strategy, you've heard about it, right? We cannot run trials where we're enrolling all comers, fingers crossed, hope we get a response, and let's see if we can deconvolute in the clinic. You can't deconvolute in the clinic. You have to come in with a bet, make the bet on the hypothesis, and if you bet right, you've won, and the patient has won. Najat talked about real-world data. We're moving away from control arms, or we're really moving towards an effort to utilize real-world data to understand our controls. And the FDA has bought in on that. So what does that translate into? So instead of running a 3-arm study, now you can run a 2-arm study. What patient wants to be randomized to the control arm? I don't. Doc, give me the investigational drug, right? So now we can use real-world data to build, synthetic control arms, which will translate into getting data faster. Najat talked about trial startup. The current model and where the industry is trending is really having networks using just-in-time. And then lastly, enrollment, right? We need to identify patients months before the trial is even started. So when the trial is started, both the site and the patients have options. So what I tried to communicate to you, at least on these last few slides, is the clinical development team is desperately trying to keep up with the rest of the company. And I think where we will see tremendous strides is being able to have study startup that is in a few months rather than half a year. So with that, I will stop. I appreciate your time.